All right, so now let's talk about part 2. We are talking about cellular division. We're talking about making copies of DNA. We talked about the structure of DNA. So let's talk about now how we're going to make copies of that DNA, or DNA replication, or DNA synthesis. And so let's pretend that I want to make an exact copy of a chromosome. Like, why would I want to do that? Well, you know, if a cell is going to divide, it needs exact copies of every chromosome in each of the daughter cells. And so you need to take the existing DNA and make an exact copy of it. There's another reason why too. You know, now that we know how cells do it, we can do that in a lab. So let's say that I've got a piece of DNA and a very small, tiny amount of DNA in a lab, and I want to make lots of copies of it. And so when does that happen? Well, it happens all the time. So for example, it's a crime scene, you know, and if you're swabbing for DNA at a crime scene, you're not going to get a lot of DNA. It's very hard to analyze if you only got a little bit of DNA. But if you could make a whole bunch of copies of that DNA, well, now that's a lot easier to work with. And it doesn't have to be a crime scene. Anytime people that work with DNA, now that once we figured out how DNA makes copies of itself, or how the cell makes copies of DNA, we learned how to replicate that in the lab. And so you can take a piece of DNA and you can make just a gazillion copies of it. And so then you can work with it. It's much easier to work with in the lab. So that's another reason why we want to make a copy of DNA. Okay, so how are we going to do this? How does the cell do it, or how do we do it in a lab? Chromosome has 2 strands, right? I can rip those strands apart, and then I can build new strands from those existing strands. And in that process, I'll have an exact copy. And how can I build a new strand? Well, I know, if I know one strand, I can figure out the other strand, because A always pairs with T and C always pairs with G. Okay, so here's another little example. Here you've got the parent molecule, you've got the DNA, you know, the piece of chromosome that you want to replicate. And so we pull those strands apart. And so again, here's one of those strands. If I've got the one strand, what's the complementary strand of that piece of DNA? And just like before, A is always with T, C is always with G. And so you can figure out, again, back up here a second, you can figure out what is the complementary strand to that piece of DNA. And so here in step C, you can see, you know, on the left, we've made that complementary strand. But we can do the same thing with the other strand of DNA. We can make its complement. And so when we're done, now we've got 2 pieces of chromosome that are identical, and are identical to the one we started with. And that's DNA replication. We started with a single DNA molecule, and we ended up with 2 exact copies of that molecule. And so the whole idea is, because we know what nucleotides pair together, we can always do this. So here's a figure from your book, again, kind of just showing a broad overview of this, showing how that one DNA molecule is getting separated, and each of those strands is used to build a new strand, and in the end, you've got 2 exact copies. And so what they're trying to show you on the right here is some of the important players in this play, some of the important things that are involved with this step. And DNA polymerase is an enzyme that's very important to this. So you remember what an enzyme is, right? An enzyme is a protein, and proteins help chemical reactions. They're also called catalysts. They help, they just help reactions go faster. And so all those chemical reactions that make you, that make a, that must go on to make a living thing be alive, all those reactions need to be helped along, and enzymes do that. And these enzymes are made of protein. And so this is why proteins are so important. So DNA polymerase is an example of an enzyme, and what's its job? Its job is to read one DNA strand and build a new strand based upon the existing strand. As always, you remember, DNA makes RNA, makes protein. Often, the protein that RNA makes is an enzyme. There's a million different enzymes that organisms need. And big picture, this is how DNA runs the show. You know that DNA is important to everything in a living organism. Well, this is how DNA does its job, right? A gene on that DNA, on that chromosome, makes some RNA. That RNA has the instructions for making an enzyme. That enzyme catalyzes some important biochemical reaction in your body. So again, this is why, a biology class, we started off talking chemistry. Living things are just a bunch of coordinated chemical reactions. And it's these enzymes that help these chemical reactions proceed faster. And so back to this figure, this is an example of an enzyme doing its work in the cell. What reaction does it catalyze? It brings in new nucleotides and it synthesizes a complementary strand by reading an existing strand. That's what DNA polymerase does. It builds new chromosomes, but there's all kinds of different enzymes in your body for all kinds of different reactions. Now, kind of leads us off to another topic here, DNA repair. So what do we call it when the DNA changes? That's a mutation. So again, mutation is a word that you've probably heard before. You've heard of a mutant or a mutation, and maybe you realized it had something to do with DNA. Usually it's considered not a good thing, but sometimes it is a good thing. But from a biological perspective, the definition I want you to know is a change in the DNA. That's called a mutation. And there's all kinds of ways that DNA can change, but just if the DNA changes, that's a mutation. Now, when this DNA polymerase is making that new strand of DNA, it sort of proofreads. It's sort of, it's bringing in the proper matching nucleotide. And sometimes there's a mistake, or sometimes, there's a mutation, but the polymerase catches it and fixes it. However, it's not perfect. And sometimes a mistake gets made and the DNA actually gets changed, and that's a mutation. And so if the DNA is changed, then the RNA that comes from that DNA, that RNA is also going to be changed. If that RNA is changed, that may or may not change the resulting protein. And so that's important to understand, because you understand, DNA makes RNA makes protein. Well, if the DNA changes, the RNA is for sure going to change, and it may or may not change the protein, but that's going to have implications for the entire organism. And so here's just an example of what I'm talking about. So here we've got a strand of DNA. It's like a new strand of DNA, right? We're replicating DNA, and there's a mistake. There's an incorrect nucleotide. So A is supposed to be with T, and C is supposed to be with G. But somewhere we got, an A with a G, or an A with an A, or a C with a T, or something that's not supposed to happen. Now, most of the time, DNA polymerase or nuclease, other enzymes catch these mistakes and fix them and pull out the wrong nucleotide and stick the right one in. And so that again helps you to make exact copies of every chromosome. But every once in a while, they don't get fixed correctly. and that's a mutation. Okay, let me give you an example of one way this could happen. Let's pretend that you're making a complementary strand of a piece of DNA. So here I've got a single strand of DNA. I need to make its complement, right? What would it be? So in your mind, what nucleotides would make up the complementary strand? A's with T's, C's with G's, right? And so DNA always has two strands. And so if we were building, copying this DNA, we would have the complementary strand, and you see how the nucleotides are paired up, right? If you only have one strand, you can easily build the second strand, and that's how you can make exact copies of your chromosomes, what we've been talking about. Now, pretend that a mistake is made while the DNA is being replicated. So, these are just chemical reactions, and so the wrong nucleotide gets stuck in the wrong spot. And so, here's an example, right? At this one base pair, we've got a T when we should have a C there, right? That gets accidentally stuck in. And so that's the wrong nucleotide. And so these enzymes are going to fix that. We need to cut that out and fix it. And that happens most of the time. And so usually those mistakes get fixed properly. You cut out the incorrect nucleotide, you put the correct one in. But what if, for example, you fix the wrong base? What if instead of getting rid of the T, we got rid of the G? And so now we have the right nucleotide pairs. We've got an A with a T, but we fixed the error, but we replaced the wrong nucleotide. We've just changed the DNA. We've just caused a mutation. And so just to, again, just illustrate what happened here, to the left is the original chromosome, right? And to the right is the new chromosome, where we tried, there was a mistake, and in fixing the mistake, we fixed it the wrong way. And you can see how that new chromosome is a little bit different. But that's a mutation. And if you change that DNA, then the resulting RNA is going to change, and that may or may not change the protein. So that's just one way that DNA can mutate. So again, what happens when the DNA mutates? The RNA changes, and maybe the protein changes. And so maybe the protein changes for the better. You get a little bit different protein, but it works better at catalyzing its reaction. Or maybe it catalyzes a different reaction, right? And that's possible, although that's rare. Maybe the protein stays the same. Even though you change the DNA and you change the RNA, it's possible that it has no effect, that the protein works just the same. That's a silent mutation, and that is very common. Maybe the protein gets worse, and that's probably the most common thing, right? So these proteins are a string of amino acids, and if you change, remember that the string of amino acids determines the three-dimensional shape of the protein, and the shape of the protein is really important to the function of the protein. And so if you change an amino acid, it's very likely you're going to change the shape, and it's very likely that's going to be have a negative effect on the protein. And so that's, I think that's why when you hear the word mutation, it has a negative connotation, because very often a mutation is going to cause a misfolded protein, and that's going to be trouble, right? Those proteins are very delicate, and they've got, billions of years of evolution to get them into a particular shape to do a particular function. And so you change them, chances are it's going to be for the worse. And so that's why I think mutation has a negative connotation. But it doesn't have to. Mutations can potentially form new, better traits. It just doesn't happen as often. Think of the X-Men, right? Where did you get the X-Men? So mutations might have no effect, or they might things make a little bit better. Often they make things worse. And so that's why mutation is important, but it's incredibly important to living things, and it's incredibly important to evolution. Okay, so why does the DNA mutate? We showed sort of one way that it can mutate. There's all kinds of ways it can mutate, but why? Why do you have these mutations? Well, I mean, there's lots of ways to answer this question, and a broad way to answer it is, cells need to divide. You just have to make copies of your cells in order to be alive. So DNA replication happens a lot. And so it's going on all the time. And like with humans, you've got, 3 billion base pairs. So there's lots of opportunity for mutations to take place. And so just because you've got so many base pairs and they're replicating all the time, chances are that it's just going to happen. You've got that many base pairs, but you've got about 25,000 genes or so. And so there's many opportunities for mutation again, right? There's many places where a mutation could cause a change in a protein. Of course, lots of mutations are caused by things in your environment, things that you're exposed to. And so carcinogens, a lot of carcinogens are carcinogenic because they damage the DNA. And there's lots of these things. And so the more an organism is exposed to these chemicals, the more chance that the DNA is going to get damaged. And so that's what these carcinogens do, is they go and they damage DNA and they cause the DNA to change, and that's a mutation. The longer you live, the more you're exposed to these things. And so that's just another reason why mutations can happen. And of course, many people purposely expose themselves to carcinogens, right? Smoking is a classic example. And so that's also something that can cause the DNA to mutate. And some mutations are inherited from your parents. And so there are some, you get all your DNA from your parents. So if your parents have a mutation, there's a chance that you're going to get that mutation as well. So there's lots of explanations for DNA mutations. Bottom line is, if that mutation happens in the wrong way, at the wrong place, it can be disastrous for the organism. Every once in a while, it actually creates a new, better trait, but more often it's disastrous. And so that's why we're concerned with them. And one good example of this is cancer. So this is why we're going to talk about cancer, because it's related to DNA mutations and it's related to cellular division. So we started by talking about mitosis and making new cells, right? And again, why do we make new cells? If you're growing, you have to make new cells. All of us started off as a single cell, and now we're made of 30 trillion, right? So to do that, you've got to divide those cells. But again, you also have damaged cells. You're also losing cells all the time. You've got to replace those old cells. So cellular division has to occur all the time in a multicellular organism. But you do reach a point where you don't need new cells. And so, once you've grown to adulthood, you don't grow anymore. You don't have to produce new cells. And there's a lot of places where, your old cells aren't damaged or they don't get damaged very much. And so, you need cellular division, but you don't always need it. And so when you don't need those new cells, then cellular division stops. And you've got genes that make proteins or enzymes which control cellular division, just like everything else. It's just chemical reactions. And so there are genes whose whole job is to just turn on mitosis when you need it and turn off mitosis when you don't need it. But if something happens to those genes, if there's a mutation in those genes that control cellular division, then it's possible that cellular division does not stop. It's possible that mitosis gets turned on and it stays on. And if you have uncontrolled cellular division, that's what cancer is. And so, of course, this is a topic that most of you are familiar with, right? You hear about cancer. You know, I think it's probably the second leading cause of death in the United States. Unfortunately, most of us probably have personal experience with cancer. We know someone who's got cancer. Maybe some of you had cancer yourself. You know, it's a disease that's just common to all humans and to all living things. And, you know, it's going to get a lot of us. And so that's why I wanted to talk about it, because it's something that we all share or know a little bit about. So let's talk about it in a little bit more detail. But what is it? It's when cells don't stop dividing and you lose control of cellular division. And so this is just kind of a figure from your book that's trying to express that. You know, you've got this tumor suppressor protein. This is a protein that, you know, and where do proteins come from? They come from RNA. Where's the RNA come from? It comes from DNA. It comes from a gene. So you've got a gene that makes this protein, and this protein's job is to suppress tumors, or to turn off or to control cellular division. But if that gene gets mutated, that can create a misshapen protein, and the protein doesn't work like it's supposed to. And so its whole job was to control cellular division, but because of that mutation, it doesn't do that. And so now you've got cells that are dividing uncontrollably. That's cancer. So let's talk about what happens with cancer. And so because these cells keep dividing, even though you don't want them to, you get a big lump of growing tissue. You get more and more cells in an area, but you don't need it. And that's what a tumor is. It's a lump of cells that are actively dividing. And so that's how you often first notice many cancers. And again, we're talking about cancer in general. There's all kinds of different cancer, and you can get it in all kinds of ways in all kinds of different places. But it's all basically uncontrolled cellular division. Well, if you get a big lump somewhere, if you feel a lump, you want to get that checked out, because that could be uncontrolled cells dividing, and so that's going to form a mass or a lump or a tumor. Now, if that tumor just stays in one place and it doesn't affect nearby tissue, it's called benign. And so a benign tumor is a non-dangerous tumor. It's cancerous. It's starting, you know, it's uncontrolled cellular division, but it's just staying in one place. So it doesn't really, you know, it might be growing somewhere that is in a bad spot, but it's not affecting other tissue. And so, you know, if you get rid of it, you should be fine. Okay, so now if that tumor starts to infiltrate nearby tissue, that's when it's called malignant. And that's when you start to have trouble, because it's going to interfere with that other tissue and it's going to start causing problems. And then what's really troublesome is if the tumor can metastasize. And what that means is the tumor starts shedding these cancerous cells. And those cancerous cells can settle somewhere else in your body and start a new tumor. And that's when the cancer is spreading throughout your body. And that's when you've got trouble, because you often can't predict where those new tumors are going to form, and they're going to be forming, and they might form somewhere where you can't operate on them, and that's when you have a lot of concern. Remember, this whole thing started by a single cell with a mutation that meant that it started dividing uncontrollably. But it's still mitosis. It's still the process we already talked about. And so each time those cells divide, they make a copy of their DNA, and the new cells each get copies of those chromosomes, right? But remember, this is the new chromosome with the mutation. So all these cells have that mutation, and they also divide uncontrollably. And so if you metastasize and you start shedding those cells, wherever they land, they're going to start dividing uncontrollably again, and they're going to form a new tumor. And that's a problem. So the trick is to get the tumor and excise it, to remove it from the body before it becomes malignant and before it can metastasize, before it can spread throughout your body. If you get it early, then this doesn't happen, and hopefully then you don't have any more problems. And so that's what this figure is trying to show, the steps here. So this is what this figure is trying to illustrate for you here. You can see that you've got this potentially cancerous cell that starts to divide uncontrollably, forms a tumor. If that tumor doesn't invade nearby tissue, it's benign, it's not a problem, get it cut out, hopefully, usually you're okay. But if it gets into surrounding tissue, becomes malignant, now it starts to cause trouble. You can still try and cut it out, but then if it becomes metastatic and starts spreading those cells, then the tumors can show up anywhere, and that's when it's spreading, and that's when you're really worried. So how do those metastatic cancer cells, how do they move through the body? What does that mean that it can just move through the body? Well, it usually starts in the lymphatic system. And so the lymphatic system is part of your immune system. And so it takes fluid from around the cells, and it filters it and sort of returns that fluid back to the blood. But it's that filtering of the lymph that helps with the immune system. And so it helps to, those lymph nodes are where that lymph gets filtered, and then that helps to remove this cellular debris or these foreign substances, right? That's part of your immune system. So that's why if if your doctor's trying to figure out if you're sick, you got a bunch of lymph nodes right here. They're feeling for swollen lymph nodes. If your lymph nodes are swollen, that means that your immune system is capturing something and trying to get rid of it. And so these cancer cells start getting into the lymph, right? Because the lymph surrounds all the cells. And so the lymph node filters them out, right? And so it tries to filter them out, but if it doesn't filter them out, that lymph gets returned to the blood, and so it can return the cancerous cell to the blood, and then of course from the blood it can move anywhere throughout your body. And so, here's a picture and showing you got a lot of lymph nodes all throughout your body. There's lots of different places where these lymph nodes occur. But those lymph nodes can filter out that metastatic cancerous cell. Now, it's possible that that cancerous cell then can just start growing right there in the lymph node, but a lot of times that lymph node catches those cancerous cells before they get into the bloodstream. And so that's why a lot of times if you have a tumor, they'll also remove nearby lymph nodes, because there's a good chance that those cancerous cells are in those nearby lymph nodes. If you leave them alone, they'll form a new tumor, or they'll break free and they'll go into the blood. But if you remove the tumor and remove the lymph nodes, then you got a better chance of getting all the cancerous cells. But if you don't, again, it can form a tumor, they can get into the blood, they can move anywhere in the body, and that's when tumors can form, and that's when the cancer can spread. But if you had any experience with cancer surgery, like when my dad had skin cancer, they found the tumor, they removed the tumor, but they also removed a bunch of the nearby lymph nodes, to look for cancerous cells and just as a precaution. So that gets into, again, what are some of the ways that we can treat cancer? And you've probably heard of a lot of these. Unfortunately, you might have some experience with some of these things, but now you can sort of put them in context and how do they actually treat these cancerous cells. Again, we cut out the nearby lymph nodes to try to catch the cancer cells before they move around your body. You also try and cut that tumor out. Remember, that tumor is made of cells that can't control their division. And so if you don't do anything with that tumor, it's just going to grow and grow and grow and grow. And so you need to cut that tumor out. And the longer the tumor's in the body, the more likely it is to metastasize and then spread throughout your body. So that's why it's always so important to be very aggressive. And if you feel a lump or something, to jump on it and try and get it out right away. And so that's what's kind of showing you here. It's showing you like, if you have a tumor and you cut out the tumor and some of the surrounding tissue, then you're getting out all of those cells that can't control their cellular division, and hopefully you're done. But if you cut out the tumor and it's spread too much, or you do the surgery and you don't get all of the tumor, if you leave some in, if you leave a single cell in, it's going to divide uncontrollably, and you're just going to have another tumor grow. So if you do the surgery and remove the tumor, you try to get the tumor and some surrounding tissue, so there's no question you got all of the cancerous cells. Another way to treat cancer is to try to kill those cells so they stop dividing. They can't control their own division, but if you kill the cells, well, then that stops, the mitosis, and it stops the growth of the tumor. So how do you kill a cell? Radiation. You hit it with certain wavelengths of energy, and that can break up the DNA in the cell and can kill the cell. And chemotherapy. You can use chemicals that kill the cell. Of course, those don't necessarily just kill the cancerous cells, they kill non-cancerous cells as well. Radiation is sometimes good because you can target a certain area and you're only killing cells in that area. But chemotherapy problem is that it kills all kinds of cells. And so the most susceptible cells to chemotherapy are ones that are actively dividing, like hair cells, intestine cells, white blood cells. And so that's why you get so sick when you're on chemo. You're poisoning cells, and you're trying to poison the cancer cells without poisoning too many of your other cells. But it's a delicate balance, and that's why you get so sick when you're on chemotherapy. Now, the good news is that, by studying and using the scientific method, we're learning so much about cancers, and we're able to develop new medicines that don't have all the harmful side effects. And so there's new drugs that are out there now that target the proteins made by the cancer cells. And so you're not harming non-cancerous cells, you're just harming and targeting those cancerous cells. And there's lots of different ways that you can do this. But by learning more about cancer, and by studying it, and by learning more about DNA, then you can develop these new drugs, and hopefully you can lead to cures for all these different cancers. And you have much fewer side effects as well. And so, I don't want to leave you on a downer. Cancer is a bummer, but it's a reality, and it's something that most of us are going to have some experience with. So it's good to understand what that actually is. But again, I want to leave you with hope, is that there's lots of advances being made, and there's this is why we need people in biology. This is why we need people to come to college to learn biology and to go to med school, because this is these diseases are going to affect all of us, and by studying them, we're starting to learn new treatments that can hopefully wipe out some of these cancers. And I think you're going to see this in your lifetime. This is going to be much more commonplace, that there are certain cancers that we're going to have good cures for, right? But we need that science. We need to study them. We need people like you to learn about these things and to figure them out. So, anyway, we talked about a lot here. We talked about cellular division. We talked about DNA replication. We talked about cancer. So as always, let me know if you've got any questions. That's all I have for right now, and I'll see you later.